everybody. My name is Liana Burkhart. I'm an assistant professor in the plant science department. It's my pleasure to present this plaque to our
So cool.
speaker today. A little bit of history on Dr. Jingqiu. Yeah. Is that she's an assistant professor at Virginia Tech at the moment in the civil engineer and environmental engineering department. She has past training at both Columbia and Cornell. And one of the things, in really diverse systems. So I think one of the real hallmarks of Dr. Liao's work is that she really works across tons of systems, from kind of listeria and soils and vaginas and all sorts of things, thinking about microbiomes and really, really diverse systems. And we first met when she came to the Microbiome Center Symposium, oh gosh, 4 years ago? Was it 5?
When I started, like 2022.
2022, that I was organizing, and she was one of our award-winning speakers at the end of her postdoc for the talk that she gave there. And a few things that you could ask Dr. Liao about her interests, if you're interested when you meet with her, is ukulele and piano and photography are among her additional interests. So with that, we are so glad that you're here. I'm really excited to hear about one of the many kind of topics that we've done.
Thank you so much. Thank you, Liana, for the nice introduction and for hosting me. I'm very, very happy to be here today. You know, there are many, many places in the world that I will never have a chance to visit for my whole life. And also some that I visit once and never come back. But Penn State is different. Like over the past 6 years, this is already my 4th time visiting here. I definitely feel there's some special connections between this place and me. So great. Okay, so today I will be talking about the ecological process that shape soil bacterial community structure across terrestrial ecosystems, right? So the diversity, the composition of bacterial community that we observe in the soil environment, are they meant to be like that, or it's just by chance? Okay, so
oh, you sometimes have to click once on it. Yeah.
Okay. So one thing about microbes that I really appreciate is their diversity. Right? Imagine like in the Yellowstone National Park in the hot springs, very likely that you will find the Thermus aquaticus, which can withstand very high temperature. They have very powerful DNA polymerase. And if in the forest soils, the microbial composition is different, right? So you may find you see Laginibacter there, which are very good at like decomposing cellulose and other biopolymers, which are very rich like in the forest soil. And deep in the marine sediments, where the organic matter are very poor, there's some very tough microbes surviving there, like Crococydiophis. Okay? And in the marathon runners, there's some studies find that they harbor, like they got microbiome, like harbor some microbes that non-athletes have, like Valonella atypica, which can enhance the athletic performance by helping like metabolize some the practice, like exercise-induced lactase. Okay, so by the way, I really love microbes, but sometimes I really hate, their names. I hope that they have to get some like easier name for us to pronounce and remember. Okay, so microbes are literally like everywhere, right? So why we care so much about the microbes in the soil environments, especially at the system, like the community level. So they are the engineers of the ecosystem functions, right? So for example, they can drive the biochemical cycling, they can promote plant growth, they can also suppress disease. I think there's a very recent paper, maybe published early this week, like in the Cell, Host and Microbiome Journal. They found a very strong negative association between the soil bacterial diversity and the abundance of the human pathogens in the soil environment. Right? And also it's help the stabilize the ecosystem, right? They buffer disturbance and help ecosystems recover after stress and environmental disturbance. And so microbes are now born equal, right? So they are different in many ways, including ecological traits. Here, I want to highlight two dimensions of the ecological traits. One is defined by the habitat range, right? So some microbes, they occupy a very narrow habitat range, like niche range. We call them like specialists. While some occupies more places, right? Like have a broader habitat range. We call them generalists. And another dimension that we can classify soil microbes is based on their abundance, right? So some microbes are more abundant in the environment. We call them like abundant taxa, while some have way lower abundance, like rare taxa. All right, so when we take the spatial factor like into consideration, there's some interesting biogeographic patterns can emerge, right? So one very interesting and well-studied pattern is called a distance decay relationship, right? So if you look at the plot on the left, where they have like different microbial communities, right? Like in different locations, you can see their composition here very different. Right? So when they have a distant decay relationship, this is something that you would observe. Versus when there's no distant decay relationship, like across those geographic locations, their composition like more similar. Right? And when we quantify this relationship between the spatial distance and the compositional similarity of the community, when there is a distant decay relationship, we can see there is the steep like slope here. With the increase of the spatial distance, the microbial composition gets more and more dissimilar, right? So it will drop. And when there's no distant decay relationship, the line is just flat, right? So it's the similarity of the composition, like microbial composition, doesn't change with the spatial distance. And there's some like ecological factors that can drive this pattern, like selection or drift, that can make the slope like more steep, versus dispersal, that's a movement across locations. They can make the composition more similar. And now I'll talk more about those ecological processes. So in this field, there are two like opposite ecological theories are trying to explain how the microbial community like structured, how they are assembled, so that they have the diversity and composition that we observed in the environment. Right? So one is niche-based, right? So we call it, we consider that the deterministic process, they play the primary role in shaping the microbial communities. Right? So Bas Backing, he is a famous like Dutch microbiologist, once said that everything is everywhere, but the environment selects, right? So that's the hardcore. The core of this theory is they think the environments like abiotic conditions, biotic interactions, those factors and process are selecting the microbes, like determine what microbes can survive in a given environment. Okay, and another the opposing theory is the stochastic process, like stochasticity, like everything they think is just by chance. Right? So those stochastic processes include dispersal limitation, where you have like there's limited dispersal, like movement of microbes across locations. Right? So that's dispersal limitation, and also homogenized dispersal. Right? So the microbe indeed like move around, and this process can homogenize the composition across different locations. You can imagine, right? They just get mixed. So that I also can change the microbial community structure. And the last stochastic process is called ecological drift. So that is the random like birth and death of microbes. Right. So in this field, like there is a lot of debates, like arguing about which processes are more important and which are not. So why are people spending so much time like debating and trying to figure out what is the mechanisms underlying the soil microbial community assembly? A very simple answer it's important, right? It's important to understand the microbial community pattern because they can connect those to ecosystem consequence. As I mentioned, like microbial communities, like they play a very important role in the ecosystem surveys and function. And specifically, this allows to review like ecological rules, like explain the diversity and composition that we see in the real world, right? So just the knowledge itself is beautiful enough, right? Like Bacon said, knowledge is power, right? So also like allow us to predict ecosystem functions, right? So for example, how like a nutrient cycling, like carbon storage, like plant support, that we can help like clarify those processes like under different environmental conditions, and also help like forest resilience and the change. Right, so let's say if we know like the percentage of the microbial community that is determined by deterministic process, that which means this part is predictable, right? So we can predict if there is environmental change, how the microbes can change at the same time, and then what is the consequence like at the ecosystem that it can bring. And then the last is a more like practical importance is it can help like guide interventions, right? Help like engineer the microbiome for different purposes like in agriculture, restoration, bioremediation, and soil health engineering, etc. Right, so do we know everything about the microcommunity assembling mechanisms? Of course not, right? So there are knowledge gaps that otherwise there's no meaning that I'm standing here like sharing those research. So the understanding of soil microbial community assembling mechanism is incomplete. For example, many studies nowadays I have done like at like local or regional scales, the large scale distributions, the biogeographic pattern that we see, which ecological processes are more important, is not complete. There's still some like large scale studies in this space, but leveraging those like public database, some samples doesn't have very good like metadata, like environmental data connected, so that kind of like lose the ecological context, like the real like true ecological context. And then also, as I said, the microbes, they differ in ecological traits, right? How the different like ecotypes, the mechanisms, the assembly mechanism differ, limited understanding. And also soil, there are different types of soils, like in different terrestrial system, right? There are forests, like shrubland and wetland. How the microbes are assembled in those different terrestrial systems, it's still in the soil, but a different terrestrial ecosystem, how that differ, we still have limited understanding. So to fill those knowledge gaps, we conducted a very large scale soil sampling and in the natural environments across United States in 2028. So we divided the contiguous US into 14 semi-equal size sampling grades shown here. And within each sampling grade, we have 5 sampling areas, each being more than 20 kilometers apart. And within each sampling areas, we have 5 sampling, within each sampling areas, we have those 5 sampling sites, each being more than 200 meters apart. And at each sampling site, we collect 3 subsamples to make a composite soil samples. And so here, I want to summarize the samples and data that we got. So in total, actually, we got like more than 1,000 soil samples, but we select 622 samples for 16S RNA genes and protein sequencing. So this is a marker gene that allow us to characterize the microbial, strictly speaking, like bacterial community at the composition, right? Allow us where what they are. And so so we have like 16S data for 622 samples. This is me collecting soil samples like in the middle of nowhere, maybe Montana or North Dakota, North Dakota. Thank you so much.
Right.
You see, like it's just like so remote and no one lives there, and it's really hard to find myself. Anyway, so all right, so those soil samples like covers span 6 major terrestrial ecosystem. As you can see, like nearly half of our samples came from like forests and woodlands, and then followed by the herbaceous, wetland, shrubland, steppe, savanna, and barren. And also we have some samples that we couldn't assign ecosystem for them. Right, and with the 16S data, we identified more than 3,000 OTUs. So those are operational taxonomic OTUs. For people who are not familiar with like 16 data, like you can imagine, like think it as a species, like species level, different genotypes. So more than 3,000. Right? You may wonder, like with like more than 600, this actually number is small, given the sample size, like especially if compared to previous study. So we actually did very strict like screening to make sure the signal that we get here is like true signal can represent true OTU here. So end up with less OTUs characterized. And for those OTUs, they were classified into 32 phylum. So here, this is a phylogenetic tree, and the most like predominant phylum was like Proteobacterial, and so which is very common like in the soil. So paired with those soil samples, we have 34 environmental variables, right? So including spatial factors like latitude, longitude, altitude, the soil properties, including some the physical chemical properties, like pH, macronutrients or micronutrients, and also surrounding land use, right? So they are all from natural environments, but their surrounding like land use patterns are different. Right? And also we got the climate data, including like precipitation, temperature, and wind speed. Okay, so with this data, the first key questions that we were trying to address is understand how the soil bacteria community assembled, and especially how they differ across those ecotypes, among those ecotypes, right, including abundant taxa, rare taxa, generalists, and specialists. Right. So here, first let's look at the composition of those different ecotypes. The figure on the left showing the distribution of those ecotypes defined by the size that they occupy and the mean relative abundance. So as you can see, like for the abundant taxa here, like they have higher abundance, obviously, right? And also there's some like overlap with the generalists, but generalists is defined by the number of sites that they occupy. They occupy like more sites. And for the real taxa, here they are low in abundance. And the specialists, you can see they have a very narrow niche, like the habitat range here. So occupy a much fewer sites compared to the generalists, like these red groups here. And then when we look at the composition, like we wanted to know, okay, what like constitute those different ecotypes. We did the phylum at the phylum level. We did this like enrichment analysis to understand their composition. So here I want to draw your focus on all the dots on the right side of this dash line for the index of 2. So on the right side of those are significantly enriched, like which means like overrepresented. Right? And as you can see, for example, actino, this red dots is the generalists, abundant taxa, like they are actinobacter or like overrepresented in those two ecotypes. And for Cetobacteria, it's just the abundant taxa. And there are several here, like the bacterial dates. They were overrepresented in rare taxa. And the specialists here, we have like cyanobacteria, for example, they were overrepresented among the specialists. Right, so here recall that I talk about the decay relationship. That's a very interesting biogeographic pattern that microbial communities can exhibit. So here we look at the relationship between the geographic distance and the weighted unifrac distance. So here the distance shows the dissimilarity of microbial composition, bacterial composition here. And so in the previous slides, you see a negative slope, right? But here it's the opposite because that one is similarity. Here it's the dissimilarity. We actually should address this before we publish this paper. But anyway, now you understand this. Here, if we see a positive slope, that indicates a distant decay relationship. Okay, and also I promise you I have real data here. It just there's so many dots, make it very hard to interpret. So I just remove those dots and showing the fitted regression line here. So it's not the theoretical line. It's we actually have the data into the model. Okay, so here you can see they're color coded by the ecotypes. The gray one is all taxa, everything included. And for the real taxa, and so that's the yellow curve line here, and the specialists, you can see it's very flat, right? So we didn't see a relationship here among those two ecotypes. While for generalist and abundant taxa, with the increase of the geographic distance, they are getting more and more dissimilar in the composition, right? So for the all taxa, abundant and generous, they exhibit a distant decay relationship, while rare taxa and the specialists, they do not show this pattern. Right? And then next we want to know, okay, what are the environmental factors, key environmental factors are associated with the bacterial diversity that we observed in those samples and stratified by those ecotypes. And so the figure on the left showing a correlation between those ecotypes diversity against the 23 environmental variables that we had. As you can see that there's some environmental factors here like pH, aluminum, calcium, right? They are associated with a majority of those ecotypes, while some factors are more ecotype specific, right? So we find an example here. For example, the moisture is only associated with a generalist, right? So looks like they may have some universal abiotic factors shaping the soil bacterial diversity. pH is actually a widely known driver, right? At different even continental scales and local scale, regional scale, pH is just so important for the microbial community. And also we identified calcium and aluminum. And this evidence is further confirmed by a machine learning model where we predict the microbial diversity based on those environmental factors and identify the top 10 most predictive features. And you can see the top 3 emerging here as pH, calcium, and aluminum. Right? So here for also another thing that I forgot to mention, as you can see, abundant taxa and the generous, they have way more environmental factors are correlated compared to the real taxa and special taxa, right? So it gives us some hints or hypothesis that the environmental condition may be more important for the generalist and abundant taxa compared to the real taxa and specialists, right? And I will provide more evidence about this soon. Okay, so as I mentioned, so this the previous slides are all about abiotic conditions, and as I said, the biotic interactions are also important, right? So structure those community. So we to infer those, we construct this co-occurrence network for all the taxa first, and also we did this for different ecotypes. So here, this dot here is represent one OTUs, and they are color coded by the phylum. And the age, there are 2 colors. One is light orange, and the other one is yellow. Sorry, it's light gray, right? So that indicates positive and negative correlations, right? So while and there are also like some arguments about like it's about only about correlation. They may not really represent a true like biological interaction, but I think at least give us some like evidence or hypothesis that we can make. So surprisingly, we found like when we quantify the proportion of those like age weights, we found it showed like predominantly like positive correlations among OTUs compared to the negative ones. Actually, there's some, if you are interested, some studies are showing like trying to figure out if the competition or cooperation are more prevalent in the microbial communities, like in different environmental contexts, and in human, in also in soil, and also human plant microbiome. So how they like microbes are interacted. So based on this data, it appears that the positive interaction may be more prevalent, but again, you know, this needs like experimental validation. Right, and right, so next we are trying to understand each node here. They can play a role in this like the network, like community networks, right? So to understand each node, like they classify into different ecotypes. So to understand their importance in terms of this like the network topology, we trying to understand. We did further analysis to understand that. So here, first I want to talk about how we can read a node's importance in a network. So there are three metrics like centrology that we can measure. One is a degree. So this measures the influence of a node to its neighborhood, right? So for example, the highlighted node, like highlighted in red here, they have most direct links to other nodes. Here, the nodes, you can, it's in this context, it's an OTU, right? And betweenness, so there's another one, this, it shows like how often a node exceeds in the shortest path between others, right? So this is, they are acting as gatekeepers, right? So the nodes are highlighted in blue, they connect like two otherwise separated groups, right? So the betweenness like reflect that. And closeness, this centrology, this shows like how quickly a node can reach like different like other nodes. Right, so this is the one that highlighted in blue here. They can reach all others like in a fewer steps, right? So then we quantify those like three centrologies for different like ecotypes here. And so what we found, like for the node degree, here as you can see, the real taxa, we found it's higher degree centrology than abundant taxa, and similar to generalists and specialists. And for the betweenness, again, like real taxa, and doing a very good job here. It's even greater than abundant taxa, like all the other ecotypes, right? Like abundant taxa, generalists and specialists. And for the closeness, it's real taxa similar to generalists, but both are higher, significantly higher than abundant taxa and the specialists, right? So in summary, as you can see, the real taxa play a very important role in the community network, right? They have very strong ecological relevance to the community, even though they are rare, like they have very low abundance. Right, so, right, so next, like to have a like system, like framework to quantify, we get some hints about, you know, the importance of those environmental conditions, like deterministic process. How do we actually can quantify them? So to do that, we apply a framework established by Stan in the ecologist. I published a framework in ISME journal that allow us to quantify the importance of deterministic process and stochastic process based on the phylogenetic beta diversity. And so you can measure those like beta NTI based on beta MATD, so the observed distance compared to the non-model, and then we can calculate this index. So based on the values, we can separate the deterministic process and stochastic process. And within this deterministic process, we can further disentangle the heterogeneous and genius selection. So that is the selection make the community like more dissimilar across locations. And the homogeneous selection is the selection that make them more similar, right? And for stochastic process, recall that there are several different ecological processes, right? Like how we can actually entangle those processes. Scientists can always find a way. So there is a step two, like they develop those RC like Bray index that is based on the Bray-Curtis distance, again compared to the non-model. And using this index, we can further quantify the importance of the dispersal limitation, homogenizing dispersal, and ecological drift. Okay, so we apply this ecological framework to quantify the importance of the ecological process for all taxa and also each ecotypes. And here, this is what we found. So the figure on the left, this shows the result for the importance of ecological process for all the taxa. The red ones on the outer layer shows quantify the importance of the deterministic process versus stochastic process, right? So you can see here for the all taxa, like the whole bacterial community, deterministic processes are slightly more important than stochastic process. And for the deterministic process, it's primarily the homogeneous selection. And for the stochastic process, it's the dispersal limitation, homogenizing dispersal and drift. They are playing about like equal role here. And then when we stratify like ecotypes, we see very striking difference here. If you compare abundant taxa to rare taxa, see here abundant taxa is the primary forces here is the deterministic process, right? So, and similar for the generalists, it's the deterministic process play a way more like predominant role. And while for the real taxa and the specialists, the stochastic process are more important. This is consistent with the environmental factor like correlation analysis that we did. Remember that for the generalist and abundant taxa, we found a lot of correlations for those environmental variables. Again, for this analysis, the environmental data was now used, right? So it's very consistent findings by using different analysis here. All right, so the conclusion for this analysis is like the determinism plays a greater role in abundant taxa and generalists, while stochasticity is more important for the real taxa and the specialists. Right, and right, so the community assembling mechanism, as we can see, like differ across ecotypes, and for the soil. Based on the literature, it also differ across different environmental contexts, right? So for soil, based on our finding, it has a near balance, like assembling between the stochastic and deterministic process. And freshwater lake, this is our previous studies, actually shows the deterministic process, like selection, dominates under strong environmental filtering. And for the plastic sphere, there's some studies showing that the stochastic process dominates those like patchy plastic surfaces. Right? So you can see here, it could be like the environment dependent, right? And even like within the soil, so the next question that we ask is the soil environments, they can differ. They are in different like terrestrial ecosystems, right? How the soil microbial community assembling can differ among those terrestrial ecosystems. This is the second like question that we're trying to address. So as I said, our samples like span forest, woodland, shrubland, herbaceous, steppe, savanna, and barren, right? So we further stratified by those ecosystems to understand how the microdiversity looks and how the mechanisms could differ in their assembling. And so this map showing the bacterial diversity across the United contiguous US and color coded by the ecosystem here. And on the map, I think it's not very obvious, but when we did the statistic test on the diversity and showing on this this part on your right side, here we see the forest woodland has the highest bacterial diversity, while the poor shrubland here has the lowest diversity. Right, and then we further look at the distant K relationship for those like different ecosystems. And this time all ecosystems exhibit the distant decay relationship. As you can see here, we see a positive slope, like beautiful positive slopes here for all the ecosystems. It just here for the shrubland, and so this is a shrubland, this line for shrubland. This one is the herbaceous. You can see the slope is steeper than the other ecosystems. And so we know the bacterial communities in those two ecosystems, they have a stronger, exhibit stronger distant decay relationship compared to others. And the forest has actually the lowest distant decay relationship. We hypothesize that maybe the wildlife helps, like the dispersal, wildlife-driven dispersal, help homogenize, the community, so that across the location, so that they are not as dissimilar as other ecosystems. Right, and for the key environmental factors, again, the shrubland caught our attention. Right, so for here, you can see there's some soil properties, like variables are correlated with some ecosystem. But for shrubland, there is a lot. So this is shrubland, this column is shrubland. A lot of surrounding land use patterns are strongly correlated. And we further did the VPA analysis, which allow us to quantify the importance of different groups like soil property, geolocation, climate, and land use. Other ecosystems here all shows soil property are the most important factors you can see here. But for the shrubland, very interesting, more than half of the variation of the diversity is explained by the surrounding land use. Okay, so this analysis showing the key finding from this is the shrubland ecosystem is strongly associated with the surrounding land use patterns. All right, so after this, all our focus shift to the shrubland. I feel like it's not fair for other ecosystems, because when we get the figures and plots, the first thing that we look at is where is shrublands. And okay, so it very interesting and very happily we found that shrubland for the network community network, shrublands, the bacterial community in this ecosystem have the lowest node degree, right? And also they have lowest the betweenness, on average. And for the betweenness somehow is actually higher compared to other ecosystems. So this result shows the bacterial community in the shrubland ecosystem, they don't have a lot of direct connections among those OTUs, like bacteria. It's sparsely directly connected. And also they lack in those bridging, gatekeepers, right? So the betweenness is very low, but somehow it's still cohesive, they are indirectly connected. So it's show very interesting, the network topology compared to other ecosystems. And then we further apply this, the ecological framework to quantify the deterministic and stochastic process in the bacterial community assembly in those different ecosystems. Here, As you can see, the forests, woodlands, and barrens, they have slightly like the deterministic process are slightly more important, but overall I think it's quite balanced. And then wetland, also like relatively balanced, but stochastic process play a more important role for the bacterial community in the wetland, right? And shrubland stand out again, right? So shrubland, you can see here, it has the deterministic process plays the most important, like the proportion you can see here, it's the largest, right? So for the determinist process, the red layer here, compared to other ecosystem. For herbaceous, strap, and the savanna, deterministic process are important, but it's less than the shrubland here, right? So this is very consistent with our findings, right? Like the it's associated with shrubland associated with like many environmental factors, especially the land use. So we think those like can have like trigger pressures like selecting the bacteria community, like bacteria in the shrubland ecosystem, where the deterministic process play a more important role here. Right, so in summary, like for the comparison across ecosystems, we think that shrubland ecosystem is not doing very well, right? Like here in this ecosystem, the surrounding land use change can act as a significant like environmental pressure, and can disturb the bacterial community in this ecosystem. And this like strong, so it's undergo like strong deterministic squeeze, like selection, like environmental selection, filtering. And also, again, recall that the shrubland ecosystem have very strong distant decay relationship, which suggests there is a strong dispersal limitation, right? They just cannot move around and like stuck there. And so with this process, they are selecting some survivals, which end up with a very low diversity, right? The shrubland has the lowest diversity that we observe in our data set. And also some species adapt to this like dry and nutrient poor conditions. And also it shows a community that are less like connected compared to the other ecosystems, right? They may be, if their environmental disturbance happens, they may be very sensitive, and it may collapse, right? The community, like the network, right? So the overall the takeaways for this study is you see the assembly rules and mechanisms differ across soil bacterial ecotypes and also terrestrial ecosystem, right? So they are not the same. It's not uniform. And most importantly, here we identify some like universal factors that may be very helpful to monitor, like serving as early warning indicators of bacterial diversity change, right? By monitoring the pH, calcium, and aluminum may be helpful. And also the real taxa, right? Even though they are very real, like in abundance, they are important, like have very strong ecological relevance to the community network. So protect the invisible hubs. And then the last one is shrublands, right? So I think more attention should be drawn to these ecosystems, potentially like most like vulnerable to the environmental disturbance. So for the land use planning, right? Like how we can, if they care about those poor microbes in those environments, right? Think about how they can change their structure and composition and diversity, and some intervention like buffer the shrublands may we need some actions on that. All right, so with that, I would like to thank my group and especially Mia, Shannon, who led this study and hardly like help with a lot of like critical data analysis. Our collaborators at Virginia Tech also help with some of the analysis, and Dr. Whitman, Sean, and the sampling team. So the samples were collected when I was a PhD student at Cornell. That allow us to do this like large scale study, and the Global Change Center and for VA for funding. And thank you so much for your attention, and I'd like to take any questions.
Yep, that is your trainee. So that's perfect.
Hi. So in the shrublands, is there a difference in the plant community? Like, is there plant diversity differences in the other communities?
I
And
think
also,
it definitely. Yeah,
sorry, I was also going to ask about what is the surrounding land use around
Sorry,
these? What is the surrounding land use?
Oh, for the shrubland.
The shrublands.
Oh, yeah. So
Just one note, if you could repeat the question for the.
so we got the question about if the plant are different, like in the shrubland compared to other ecosystems.
Yeah, the
right?
diversity.
The diversity, and also if the surrounding like land use patterns for the samples collected in the shrublands. And so I think it's definitely, I'm not a plant scientist, and I would hypothesize the diversity would be, plant diversity would be very different. Like even for the plants, for microbes, they have to adapt to those dry and nutrient poor environments. It's the same situation like faced by the plants, right? So I think there definitely also slack pressure that acting on the plant species. So I would definitely hypothesize, and I think this is something like very interesting to look at. And for the land use patterns, I can show you this figures, which shows the right. So here, this last 10 variables here are the land use, like proportions of different land use. For the shrubland, it's strongly correlated with the like barren forest, like shrubland itself, and croplands. So like there's some like human, intensive like activities that can play a role, like pastures. So yeah, so they're make higher proportions. If there are higher proportions of like agricultural related activities, that could influence the bacterial communities like composition and shrub.
Hi, really fascinating talk, and it's really interesting to learn. I was interested in learning more about the questions you were focused on, the rare versus the abundant taxa, and you saw both deterministic and stochastic forces that structure these assemblies. And I'm curious, in the array of research and findings that you're able to look at, did you see any functional differences between the stochastic versus deterministic groups, or like traits that might make a microbe more deterministic versus stochastic in your study?
This is something I proposed in my as a proposal. It's a great question. Yeah, this is definitely something I want to learn more about at the functional level.
Because you have environmental stuff here. Clearly, there's a lot of
Yeah,
environmental
definitely. I totally agree.
from the microbe's perspective.
For
Yeah.
that, yes. Yeah, so that's because it's characterized by 16S, which prevents, characterize those like functional the profiles and genes profiles. So this is something I really wanted to do, and trying to convince the reviewer too of my as a proposal. Like, I hope I convince you that it's really important to understand those. Like somehow some reviewers just don't feel the same way. But anyway, allow me to do this analysis to understand like how it's also not about like ecological traits and functional traits, right? Like how the assembling like mechanism can vary, and then map back to those like
Rare taxa, especially, yeah.
Yeah, so this, great question.
Yeah, great talk. How do you normalize seasonality in your sample?
Sorry.
How do you normalize seasonality? Because this is a large scale sample set. So how do you normalize the seasonality, like differences in time of sampling?
Oh, right. So we are so this is systematic sample collection. Are trying to minimize, human like induced like bias. And so we first like at the sampling stage, like design the sampling map. We're trying to first control the influence of time, like temporal factors, because that can play a role like in those. So we did this like within a fairly like small time frame, and we have a standard like sampling kits that send out for collector to collect samples for us. We have I don't know, like 20 pages. Just like we can tell, help with the sampling, like
There's samples from Center County in here.
Oh, cool.
yeah. So thank you very much. So for the sampling, I'm trying to have we had a standard protocol sampling like heat and make sure it's sterile. And in the data analysis stage, you can see like the number of samples that we had like for different ecosystems are actually different, right? So this can introduce some bias because of the sample size. So we some analysis are sensitive to sample size. We control that. So what we did is subsampling identical number of samples and then do the comparison to make them like comparable.
Hi, great talk. I'm thinking of the positive relationships in the co-occurrence network. Did you have like a large range of richness across samples? Because I guess in my mind, if you have some that are really rich, then you have like more of your taxa in there and they're co-occurring there and that's creating those positive relationships. Is it? Yeah.
Right, so this is actually are you a reviewer of? So yeah, the one reviewer pointed out, okay, so especially the way that we classify those real taxa, and it could influence by the threshold that we picked. And so we did this actually try the different levels of like the thresholds to classify real taxa, abundant taxa, we get consistent results. And so yeah, so I think it's not like influenced by, you know, the number of taxa like assigned to each like.
Oh, great talk. So my question has to do with scale and how you sampled. And so I'm wondering, if we look at ecosystems like shrublands, for example, there are heterogeneous different types of soils and different types of shrublands, right? So for example, in California, you have maritime shrublands and chaparral environments, and then, you know, in the inner coast. And so is that something you really factored for when you were doing your original sampling, the fact that you have multiple types?
Yeah, great question. So we didn't, because for those field studies, it's really hard, especially large scale study, it's really hard to take everything into every factor into a consideration. So our approach was trying to have like even distribution of the samples so that it's not biased by the geographic location. So that was the focus. Let me see if I can show the map again. That was the focus of the sampling design. Like we separate to make sure that at this large scale, like different locations, we get some representative samples. So we didn't take that into consideration, and also we have to compromise some resolutions here, like at a large scale, right? Like at the local level, like regional levels, we have to compromise those sampling resolutions. So this is definitely some like follow-up studies to look at like those subtypes, right, of those soil. And it's the same ecosystems, shrubland, but you know the plant could be different, and the climate regions in different climate regions definitely could be different. Like it's a very nice follow-up study.
Yeah, and just that's great. I mean, of course you can't, you know, look at everything, sample everything, right? So, so I like this idea that you want to go a little deeper and look at that heterogeneity of different communities, but I would also think about maybe weighing that based on maybe the states or the regions, because some regions like California have so many subtypes, whereas others don't. And then also the confounding factors then, because like, for example, again, California, you have land use, but climate is a huge deal, where it turns out that shrublands like chaparral are actually predicted to not necessarily be vulnerable, but the forests are, and they're going to grow because it's getting warmer. So, I mean, I love this study, and I think there are a lot more questions, but just more of a comment there.
Thank you so much for the comments. Very helpful.
Okay, Jasmine, the last question.
Did you in some way correct or consider the impact of rain? Because I see in your graph, precipitation is a significant factor in multiple ecosystems. And if you think about the microbes after drought, and then you suddenly have lots of rain, that will drastically change the composition, and maybe something that was rare before is now going to predominate. So how does that impact your
Yeah.
analysis?
Very good question. So for this precipitation here, it's we are looking at the long-term like precipitation. It's a climate, right? Like 10 years precipitation that which like reflects the a long-term, like it's not about weather, right? It's a long-term effects of the climate on this. And raining definitely can influence like day by day, right? If it rain or not, it can influence. We have actually urban fecal contamination of the in urban river. The raining is the key factor, the most important factor like shaping the fecal contamination. And so to account for that, we ask the collectors, please find a sunny day for sampling to control the effects of the raining. Like we don't allow any sampling happen during the rain or right after the rain. So because that can definitely change a lot for this the findings.
Okay. Pedagogical question. Wonderful talk. You had a couple of slides that had a reference of Notebook ML. Can you explain what that is?
It's an AI tool, very popular AI tool. So I don't know, I feel like it's doing many things that it's very capable. And here I ask it help with some like the visualization. Like for example, this one, I really like it. Like this,
Yeah.
came up with this like it's assembling funnel for the environmental selection and those like random like stochastic process. And I think it did a very great job. And so I used that and I was excited.
Thank you.
Nice. Like the visualization, like illustration is great. Yeah. Thank you. So